We outline a system to assess the dynamic formation of chromatin domains. We utilize the system to follow the recruitment and spreading of PRC2-mediated chromatin domains in cells. The process can be paused at any time to analyze the events in progress. This system could be geared to monitor dynamic changes in any given cellular process, and hence is not limited to tracking chromatin domain formation.
Start by designing guide RNAs to delete exon 10 and 11 of an endogenous copy of embryonic ectoderm development, or EED, in mouse embryonic stem cells using CRISPR design tool. Clone the guide RNAs into the CRISPR-Cas9 plasmid according to manuscript directions. In a 6-well plate format, transfect 200,000 mouse embryonic stem cells, or MESCs, with 1 microgram of each guide RNA using transfection reagents according to manufacturer's instructions. Change the media 24 hours after transfection. 2 days after the transfection, isolate GFP-positive cells using FACS. Transfection efficiency ranges from about 10 to 30%, so sort around 500,000 cells to capture sufficient GFP-positive cells for plating. Plate the isolated GFP-positive cells into 15-centimeter plates that are pre-coated with 0.1% gelatin for colony picking. Grow the cells in ESC medium for about 1 week until single colonies are visible, making sure to change the media every 2 days. Pick a minimum of 48 colonies by using a 20 microliter pipette tip to scrape over the colony while aspirating. Without breaking up the colony,